First of all, thank you very much for giving me the opportunity to talk here today. I am very excited to talk about the project that has received seed funding from EHDN and the project that we hope will give novel insights into the molecular causes of Huntington's disease. Now, today I'm going to talk about splicing, and it has become increasingly clear over the last decade that splicing is deregulated in Huntington's disease. This has been found mostly also in postmortem studies, RNA sequencing studies from patients, but in addition then also in several HD mouse lines as well as cell models. Recent work from our group has, in addition, found another splicing pathway that is less known, so-called backsplicing, is altered in Huntington's disease. What is backsplicing? Backsplicing generates covalently closed RNA molecules that are extremely stable. They come in different flavors. Do you see this? Yes. They can be intronic, exonic, or a mixture of both. And again, they are extremely stable because they don't have any open endings, and they are enriched in the brain. How about their function? What do circular RNAs do? They are at this point severely understudied, but for some of them, it has been shown that they can regulate transcription in the nucleus. If they're exported to the cytoplasm, they can have a very large set of different functions that I won't go into detail. However, they can be very important, again, especially in the brain. Now, all of this made us very curious about the fact whether or not there could also be a circular RNA transcribed from the Huntington's disease gene Locus. And we investigated this by looking into a database that is called CircHTT that collects and summarizes RNA sequencing studies focusing on circular RNAs, and we found a promising candidate. It was replicated by 4 independent studies, and it is enriched in the brain. And we called it CircHTT for simplicity. CircHTT includes exons 2 to 6 of the HD gene Locus, so it does not include the CAG repeat track that we have heard so much about today. It is conserved. We later found out that this is conserved in mouse and pig, and it carries on the flanking introns these very repetitive sequences, a hallmark of circular RNAs. Now, what we think is happening is that through these repetitive sequences, they undergo intron pairing, leading to the formation of this very stable circular RNA molecule. And it can be only differentiated from its linear counterpart by using divergent primers that would point into opposite directions on the linear transcript, whereas give basically a unique amplicon over this so-called backsplice junction on the circular RNA. We furthermore were able to validate that this is the sequencing basically of this specific region. We furthermore were able to validate that this molecule indeed is circular by using RNase-R, which digests basically every other linear transcript. In case of CircHTT, it enriches the abundance of this molecule. Now, of course, we were interested in understanding where it is exactly expressed. And in human samples, we found that it is enriched in the brain, in neuronal tissues, in cerebellum and whole brain, as well as in the spine. And when we were able to make use of terminally differentiated neurons from human control and HD patients, we could find that it is increased, its levels are increased in the HD sample group. Importantly, it also seems to strongly correlate, the expression levels seem to strongly correlate with the CAG repeat length. We were excited since we could then also replicate these findings in mouse. First of all, we see this increased levels of CircHTT in brain. Especially, it seems that in the striatum we have an enrichment of CircHTT. And when we then made use of the well-studied Q175 mouse line and looked into tissue expression of CircHTT in the cortex, striatum, and cerebellum, we again observed these increased levels of CircHTT in these brain areas, as opposed to the well-characterized decreased levels of linear HTT transcripts. Now, of course, the big question now is, what does this molecule do, and is it relevant to Huntington's disease pathogenesis? We try to investigate this by using overexpression of CircHTT, so synthetic overexpression of CircHTT in cell lines. And to cut a very long story very short, we see conflicting results. First of all, we observed that it may increase levels of Huntington protein, but then we also saw that it seems to decrease Huntington protein. So in the end, we don't really know what it is doing in this case yet. Another way of tackling the question of the function of this molecule is by specifically knocking it down. So we are planning to use antisense oligonucleotides and shRNAs that specifically target the spec splice junction, the unique feature of this molecule, and perform knockdown experiments both in vitro as well as in vivo to understand its relevance biologically in general, but more specifically also for Huntington's disease. And with this, I want to thank the entire group, of course, my PI, Björkqvist, and all the people that have been working on this project so far. I just recently joined the team and very much looking forward to see what it brings. All our collaborators, of course, and our funding. Thank you.
Thank you very much. Any questions out there? Yes, we have a question. Great. You're awake.
Thank you for a really nice presentation. Can you say something more about these repetitive sequences that are pairing between intron 1 and intron 6? Is that downstream of the polyadenylation site that is used for exon 1 production?
That is a very good question. It is all over the intron, so it's not a very specific region. But when you look at this, I can't go back. If you come to my poster, I can show you that it's basically in all the regions of the intron. So it's not really specified at a certain locus. But it is an enrichment of alu sequences that in previous studies on circular RNAs have been shown to really mediate this intron pairing and then the formation of the circle. But there doesn't seem to be a very specific spot where you actually see these repetitive sequences.
Thank you.
Thank you very much. Do we have another question out there? No. Thank you to our speaker. Thank you very much.